Good afternoon, everyone. If you don't know me, my name is Dr. Colin Bird, and I supervise the CPFW Selfish Health Lab. Today, I'm very proud to introduce one of our very own, Dr. Lindsay Alma, who is a Davis EML postdoc. So for a little bit of background on Lindsay, she received her BS in 2013 from UCLA in environmental science, followed by doing several internships before starting a master's degree at Cal State Nichols Hills, before transferring to the University of Washington with her advisor, Dr. Camino, and receiving her PhD in 2021 from University of Washington School of Aquatic and Fishery Sciences. Lindsay today will be talking about unraveling health and disease of shellfish and hillgrass. Take it away.
Is this on? Can you hear me okay? Yeah, okay. Thanks, Colleen, for that intro. Again, my name is Lindsay Alma. You may not have recognized me. I'm here at the Bodega Marine Labs just visiting. I am a postdoc with the labs, but I actually live in Seattle. So that's why you haven't seen me before. But just some research history. I did my undergrad at UCLA where I studied deer and their connectivity between highways. And then I worked for the Los Angeles Department of Sanitation where I took invertebrates and I put them into sewage effluent, and we wanted to know how their physiology would change. Then I worked for NASA where I did remote sensing of sewage plumes. And then I did my PhD at University of Washington, where I studied shellfish physiology. And now I'm here at Bodega Marine Labs, where I'm studying seagrass and seagrass wasting disease. I'll be talking to you guys about these two topics, shellfish physiology and seagrass wasting disease today. So my big overarching question is, how does organismal health change with stressors and with disease? This is a really, really broad topic, and I hope to even just, in a little way, chip away at this large question. My talk is going to be split into two parts. Part one is shellfish physiology. Part two is seagrass disease. For shellfish physiology, the first species I'm going to be talking to you about is the purple hinge rock scallop, Crassodoma gigantea. They are native to the West Coast. They form these really beautiful reefs, and they're a new aquaculture product. So a lot of aquaculture industry is really interested in producing these guys for food. Then we have the Olympia oyster, Australurida. They're native to the West Coast. They are experiencing a little bit of competition with the Pacific oyster, which is the larger oyster that we eat. And there's a large restoration effort to try and replenish their populations. And then we have the Mediterranean Muscimiddle Scaloprovincialis. They are non-native to the West Coast, but they're highly cultured worldwide. They're cultured here, and they're grown usually on these things called long lines. And then in part two, I'll be talking about eelgrass, Zostera marina. They are native to the West Coast. They can stabilize sediment, and they provide 3D habitat for other organisms. And I'll be talking briefly about the Pacific oyster. That's the bigger oyster that I just mentioned, Crassostrea gigas. They're non-native to here, and they're highly cultured worldwide to eat. So part one is shellfish health. The ocean is very dynamic. It's constantly changing through time and space. Coastal dynamics are especially unique, and they're a lot more variable than the open ocean. Climate change has already begun to change the ocean chemistry. For example, this is a map of anomalies of sea surface temperature last year. And organisms can actually have the potential to change their physiology to cope with some of these environmental stressors. Now, when the general public thinks about climate change, they probably think of an image like this. But most of the climate change effects are not quite so obvious to the general public. So before we talk about shellfish, I want to lay a little bit of some background. So carbon dioxide is out in the atmosphere. When it dissolves into the ocean, it causes a warming ocean. It causes less oxygen and more acidic conditions. And this can have a negative effect on shellfish metabolism, diet, reproduction, and shell integrity. And this is just an example of climate change in action. From where I'm from in the Pacific Northwest, we had this heat wave in June 2021, where the heat got as high as 818 degrees. So that was extremely hot for everyone there and also all of the sea animals, because this actually coincided with the low tide. And the air temperature was so hot that it actually cooked these animals alive. Webs are also going to change with changing carbon dioxide emissions. As you can see, shellfish are right smack dab in the middle of the food web. So without shellfish, if they start to go because of climate change, there's going to be no charismatic megafauna like the octopus or the seal or the orca. Climate change is predicted to worsen as human population grows exponentially. So right now, our human population is at about 8 billion. In the year 2100, it's predicted to be 11 billion. So to provide a protein source for this growing population, we need to rethink and find a sustainable protein with low carbon emissions. And that's shellfish. So you can see on this graph, probably the worst meat that we can eat is beef, and the best meat that we can eat is shellfish. They're really sustainable compared to all the others. Bivalves also provide a lot of ecosystem services. They filter the water by consuming particles. Bivalves can prevent erosion and stabilize shoreline. And bivalves are a foundation species. They form these great reefs that can build habitat for other settling organisms. This is Pike Place. This is a very famous farmer's market in Seattle, where I'm from. And they sell a ton of this kind of spread of shellfish. So it's extremely economically important. It supports tourism, and it's a sustainable protein. I also noticed here in Bodega Harbor, these people are actually digging for shellfish. So it's extremely important here. People are digging for them to eat. So my research themes and questions are as follows. First, for this half of the talk. First, acclimation. This is an experiment done in the laboratory. So I want to answer, how does ocean acidification and warming affect the physiology of shellfish in the lab? And then acclimatization, which is in a natural laboratory or in the field. How does physiological performance of shellfish change in its natural environment? And then finally, parental carryover, which talks about the next generation. So we want to answer, how much does parental environment really affect the quality of the offspring? So if parents experience this environmental stress, will their offspring be better or worse off? Starting with acclimation, let's see how quickly bivalves can change their physiology with changing environments in the lab. So this first experiment was on the purple hinge rock scallop. We collected them from this oil rig in Los Angeles, California. And remember, this rock scallop is a new aquaculture product to a lot of shellfish industry leaders. This is me with the tanks back when I was young and innocent. So we had a 6-week exposure of either 14 degrees, which is ambient, or 21.5 degrees, which is what's predicted for the next century. And then our pHs were pH 8.1, which is ambient, and then pH 7.6 predicted for the next century. So we had a full factorial experiment where we had an ambient, high temperature only, acidified only, and then a multiple stressor. And I was really interested in the shell layers. I was interested to measure shell density. So shells usually have 3 layers. The top layer is the periostracum, which is the rough outer layer of the shell. The prismatic is the middle, densely packed layer. And the nacreous is that pearly layer that you usually see on the inside of shells. And what I was most interested in was the periostracum, because that's what's in direct contact with the water, and that's probably what's going to dissolve first. So to test this hypothesis, we used a micro CT scanner, and we took thousands of X-rays, stacked them all together. And what you're seeing here is a bunch of stacked shells. And on the bottom are these little calibration rods that we use to calibrate density. And here's the results. So on the x-axis, we have 14 degrees, and then our hotter 21.5 degree temperatures. And then we have our pHs of 8.1 and 7.6. And what you can see, the y-axis is periostracum density over total density. And what you can see is temperature didn't have an effect on periostracum density. The acidified conditions had a little bit of an effect, but when combined, they had an additive effect where they actually decreased the periostracum density quite a bit more. We next did some sequencing to find bacterial diversity. We wanted to know, will virulent microbes be more prevalent with climate change? And this was really important to the aquaculture industry to know. For example, there's a human illness called Vibrio that can be caught from shellfish. It can shut down farms, it can affect their profit. And some Vibrio species are completely harmless in low doses, but once they get exacerbated, they can be pathogenic. There's also Vibrio pathogens that can infect the shellfish themselves. So what I'm showing here is our 2 low temperature treatments, one at high pH, one at low pH. They have sort of a similar bacterial community. You can see that that green color sort of takes up most of the bacterial community, and the sort of medium blue color there as well. But when I pop up the high temperature treatments, we see a completely different bacterial community. We're seeing that this pink section is actually dominant. And if you look at the legend, you can see that that's actually Vibrio. So that was really, really surprising. So that means that climate change-related stressors do, in fact, change the microbial community and has the potential to encourage Vibrio and some other dangerous pathogens to concerning levels. So next we want to test our findings in the field. I'll introduce to you our 2 species that we worked with in the field. The first one was the Olympia oyster. They had a crash because of overharvesting in the 1920s. You're looking at, in the red is Puget Sound, which is in Washington. That's the study system that I used. So they were overharvested, they never recovered. There's a lot of restoration, I know, here in California, in Washington as well, to try and replenish some of their populations. And I just wanted to show you the difference between a Pacific oyster and an Olympia oyster. So the Pacific oyster is a lot larger. They're non-native and they're quite a bit more resilient. So they'll be able to withstand some of these conditions and also outcompete the Olympia oyster for space. And these are the long lines that I mentioned. This is what mussels are usually grown on. And you can only imagine that the quality of the product is going to vary based on where they put the long lines and how deep the long lines are. This is Puget Sound. It has a very complex geography. It was carved out by glacial ice during the last ice age. And our study sites are Hood Canal, Central Sound, and South Sound. And these all kind of are in the realm of the greater Seattle area. And we use these things called orca buoys. We were really lucky to use these oceanic remote chemical analyzers, where we actually attached our shellfish physically to the orca buoys, so we could see exactly what was happening to them 24/7, because these orca buoys have very high resolution oceanographic data that they were constantly recording at depth and at different sites. So we put our shellfish underneath the orca buoys. I'm going to use the same colors throughout this presentation to represent the 4 study sites that we have. So at South Sound, we have a cage at 5 meters and then a cage at 20 meters. In Hood Canal, we have a cage at 5 meters, and in Central Sound, we have a cage at 5 meters. We did a 1-year outplant starting in July of 2018. We collected them 6 months in and then 1 year into the experiment. And we had some engineers create these really lovely cages for us. It made out of really strong PVC and has a lot of reinforcements. But the sad part of it is that they sank and fell into the abyss, and we are very sad. But the show must go on. This is us just out at the buoys. We had to scare away some sea lions in order to get to our buoys. And this is our dive team. This is some underwater footage. So this is the one on the far left is the package that is underneath the orca buoy. So that thing actually goes up and down with time and is able to record all of the oceanographic conditions. And the picture on the far right, that is what I came up with after our very poorly engineered cages sank. And this is a very scientific thing that I bought on Amazon. It's a raccoon cage. And it actually worked really, really well. So these are some box plots of the oceanographic conditions. The black center of each box and whisker is the median. I also put the mean in a white dash. So there's endless patterns because it's a super complex system. And I don't have time to go through them all, but I'll point out some obvious trends. Like right here, you can see that Hood Canal in orange has a very wide variation of temperature. And South Sound at 20 meters, that was the deep site, has very low variation compared to all the others, especially compared to the South Sound at 5 meters. They're only separated by 15 meters, and they had vastly different oceanographic outcomes. And this is salinity. So you can see that the orange, the Hood Canal, is has a much lower salinity than all the others. So there's probably a lot of freshwater input that's going into it. And then I'm showing the pH plot and then the dissolved oxygen plot. We think that there's higher upwelling during the first half of our experiment. You can see that the pH and the dissolved oxygen is higher in the second half of our experiment. And then this is chlorophyll. You can see that both South Sound at 5 meters had quite a bit of chlorophyll. And that's really interesting compared to South Sound at 20 meters, the green one. So there's some kind of stratification there where the phytoplankton is not getting down to that 20-meter range. And then this is aragonite saturation. So anything below 1 is considered corrosive to shellfish. And we can see in some instances it did get below 1. So that's of concern for us as well. This is growth rate of the Olympia oyster after this 1-year outplant. On the x-axis, we have our sites. On the y-axis, we have growth rate. And then the two panels are separated by our time 1 and our time 2. Time 1 is July to January, and time 2 is January to July. And as you can see, both site and time affected growth. The South Sound sites had really much different growths from each other. And again, they're only separated by 15 meters in both cases. And that was the case for mussels in time 1. But in time 2, there were no differences in growth between the mussels. And we think that that's because in time 2, the water was a lot more well mixed. And then in time 1, it was a lot more stratified. This is a hydraulic press. You guys maybe seen some videos of this on like Facebook where they press like random things. Yes. So we're pressing shells. We're poking holes in shells to see how strong the shells are, to test for, to see if the ocean acidification affected the shells at all. So on the x-axis, we have the same axis as the last graphs. On the y-axis, we have shell strength measured in newtons. And again, they're separated by our 2 time periods. And there's a clear difference in shell strength between collection times. Between the 2 collection times. But unlike growth rates, shell strength didn't really matter between sites. So they had pretty much the same shell strength amongst sites. That wasn't the case for the mussels. We see that, again, the South Sound at 20 meters had much lower shell strength. They had the most undersaturation of aragonite in the whole experiment. But the rest of the sites were the same. Same thing with the second time period. South Sound at 20 meters, much lower shell strength. The rest were basically the same. So this suggests that there's a difference in species of shell strength, possibly differences in shell composition. We're thinking that maybe since the Olympia oyster is a native species, they're able to cope a little bit better with some of that low acidity, and the non-native mussels are not used to that, so they might have worse shell strength because of that. So this is an isospace biplot. It looks complicated, but I'll explain it as best as I can. We're measuring nitrogen-15 and carbon-13 isotopes. On the x-axis is carbon-13, and the higher you get in carbon-13, the more marine your diet is. And then y-axis, we have nitrogen-15. So the higher nitrogen-15 you have, the higher trophic level you are. And what we have here is the end members. So our first end member is a terrestrial end member. An end member basically means a diet source. Terrestrial, that means that there's little particulates from the land that are getting into the ocean, and the shellfish are eating them. Phytoplankton is their main source of food, so we're going to see that the signatures are going to be kind of revolved around that phytoplankton end member. And they might also be eating macroalgae, particulate organic matter, maybe even some zooplankton. This is what our signatures look like. Bless you. You can see that they kind of break out into 2 separate cohorts, where the Hood Canal and the Central Sound are sort of skewed to the left, and the South Sound sites are skewed to the right. So what I'm thinking is that, remember, there was a lot of low salinity in those 2 sites. So what I'm thinking is there's a lot of runoff into the ocean, and they're sort of just consuming any of the particulates that came off the terrestrial environment. And then the oysters from the South Sound, they might be consuming more zooplankton, more particulate organic matter from animals. And there also might be some upwelling there that might be changing the signature of the phytoplankton in order to increase its nitrogen-15 level. Now talking about parental carryover. So does parental environmental history affect offspring performance? So I'm going to be talking about the Olympia oyster here and their early life stages. It's really important for them to be able to tolerate the changing oceans at that early life stage because we want to be able to create breeding programs to create a more tolerant line of oysters. So the Olympia oyster life cycle is sort of strange compared to some of the other invertebrates. They're hermaphroditic. The males will sperm cast into the ocean just like any other broadcast spawner would. And then the females will collect that sperm and actually brood the larvae in their little brood pouches for 2 weeks, and they'll release them as veligers, fully formed swimming veligers. They'll turn into petaveligers, they'll settle as juveniles, and the process will start over again. So we used our Olympia oysters from the previous experiment, and we spawned each parental cohort to make 4 larval groups based on parental acclimatization. So we have our 4 larval groups. Then we challenged them to either 14 or 20 degree temperatures for 2 weeks. We worked with a nonprofit here called Puget Sound Restoration Fund, and we wanted to help inform them on their restoration efforts of the Olympia. So for some results, on the x-axis we have our parental locations, so the parents of the larvae, where they came from. On the y-axis, we have growth rate. And what you're seeing here is the growth rate of the oysters at 14 degrees Celsius. There's not a whole ton of difference between the populations. But then when we pop up the 20-degree one, you can see that there's quite a bit of a difference. You can see that the South Sound at 20 meters just couldn't keep up with the others. And it's really interesting because this actually reflects perfectly the parental growth patterns. That means that there probably is evidence of parental carryover right there that has allowed the larvae to sustain some high temperatures. So conclusions for this half. Plasticity of shell properties. There was plasticity of shell properties, microbiome growth, and diet with changing environments. Shellfish grown on long lines might have differences in product depending on water stratification and location. We saw that there was a thermotolerant phenotype that carried on to the offspring to make them grow larger, faster. And this all has implications for the aquaculture industry and restoration breeding programs to withstand climate change. That's the end of the first half. Now I'm going to be talking about seagrass health. This is the research that I've been doing over the last 2 years here at the Marine Labs, and yeah. So seagrasses also have some ecosystem services, just like the bivalves. They help with fisheries by providing a nursery for juvenile fish. They also create a 3D habitat that allows other organisms to live. They're ecosystem engineers. They prevent erosion. They also sequester carbon and reduce ocean acidification. But eelgrass beds are disappearing due to environmental change and disease, so we're really concerned about that. I'll be talking to you guys about seagrass wasting disease, which is the disease that I study. It's caused by Labyrinthia zostra, but from now on I'm just going to call it Labby because that's way easier. It's a single-celled protist. It looks something like this under the microscope. And it's related to a slime mold. So you may have seen something like this on your hike. It's very closely related to the terrestrial version of a slime mold. It occurs naturally. It infects Zostera marina. Zostera marina is the most prominent temperate seagrass species, which is in huge decline right now. And Labby creates this dark necrotic tissue and loss of seagrass beds. And we're concerned that it may be spreading anthropogenically as well. This is something called the disease triangle. You need 3 things to have disease. You need a susceptible host, that's the eelgrass. You need a pathogen, that's the Labby. And then you need a conductive environment. And we're not entirely sure what that conductive environment is, but we're trying to run experiments in order to answer some of those questions. So the research themes and questions for this half are an experiment with oysters and eelgrass. Will oyster sanitization methods reduce disease? With behavior, can we identify cell-level Labby eelgrass interactions through microscopy? And then water residence time. What is the water residence time of Labby? First, we'll go into oysters and eelgrass. Oysters and eelgrass actually share a lot in common. I'll be talking about the aquaculture product Crassostrea gigas. This is the one that you would eat. I've heard that Tomales Bay makes a lot of great oysters. But bivalve beds also co-occur with eelgrass beds quite often. There's evidence that oysters can reduce Labby by filtering it, but they may also transmit it. And then they may spread via bivalve seed transplant. So basically, a lot of these shellfish farms will create little baby oysters, and they'll ship them out to other places. And it's possible that Labby might be living on those oysters, and once they're transferred to other places, they could infect the eelgrass that is there. So what are some sanitization methods that we can do to try and clean off those oysters before they get shipped off to other places? We could give them a really dilute bleach bath. We could put them in a low salinity bath. Or we could depurate them, which means putting them in super clean filtered water and letting them kind of clean themselves out. This was our experiment. It was done in Washington State. Over at Friday Harbor Labs, we inoculated our oysters. We had either live oysters or just oyster shells, because we wanted to see if the live oysters were actually filtering out the Labby. So we inoculated half of them, didn't inoculate the other half. And then we gave them either no treatment, very dilute bleach baths, 10% salinity bath, or a 24-hour depuration. And this is what one of our tanks looked like. You can see the oysters on the right in a little baggie, and the eelgrass that you're seeing just right in the middle has a really, really intense lesion from seagrass wasting disease, the black part of the seagrass. This is what our experiment looked like. There were 80 tanks. It was quite a feat to run this experiment. And this is in the Friday Harbor Lab, Ocean Acidification Lab. To measure disease severity, we actually took our seagrass and we put them on a scanner, and we scanned them into the computer. And then we used ImageJ to trace the diseased area and then to trace the total area, and that dividing the disease area by the total area will give us severity. These are our results. On the x-axis, we have the oyster sanitization treatment, either a control, no treatment, bleach, low salinity, or depurated. And then on the y-axis, we have severity. The colors are, if they're blue, there's no oysters. Orange has live oysters, and oyster shells are in green. And what we found was there was no significance between live and dead oysters, but there was a significance between sanitization methods. So you can see with no treatment, with no sanitization methods, the severity was quite a bit higher. But with sanitization methods, it was quite a bit lower. But you may see that in our control, we actually did have some disease, and that might be a confounding variable. We had just some background infection that we couldn't get rid of, and that's why we have disease in our control. We also want to point out that we inoculated them with a much lower dose of LABI than we usually have because we wanted it to be as close as possible to environmental levels. So that's why we didn't see some disease in some places and saw disease in other places, and why we had some background infection. So the next steps for this experiment, I'm going to be extracting DNA from eelgrass, from oyster gut and gill tissue, from swabs that we swabbed on the oyster shells. From water, we're going to extract eDNA and RNA. And then oyster histology, we want to see if we can find the LABI within the oyster tissue. Next is microscopy. We did this in collaboration with Bigelow Labs in Maine. And I want to thank John and Maya for helping with this part of the presentation. So this is LABI. This is a big clump of LABI and a bunch of them just kind of going in and out of this big clump. This is how they behave. They have these like slime nets that they're using as kind of highways. This is how they behave in seawater. This is how they behave in a nutrient-rich broth. This is a healthy eelgrass. We are illuminating it with a red fluorescence, which fluoresces the chlorophyll. And this is a sick piece of eelgrass. So this is a leaf, and then this is the lesion that we're seeing. So there's no chlorophyll in the lesion. We use this green stain, which is called phalloidin, which is a mushroom-based stain that stains the slime networks of the LABI and stains the F-actin. So you can see that the LABI is starting to get into the chlorophyll cells, and they're spreading these little tendrils and making these pathways in order to get deeper into the eelgrass. And this is a time lapse of infection. The blue are live seagrass cells, and the black are dead seagrass cells. And this is what happened overnight. Yeah, they're creating lesions. This is one of my favorite ones. We can see that the LABI is actually making this kind of staircase type structure. And that's what you're seeing in the leaf that's right there, the kind of line of LABI there. That's really interesting. And this is just another one of them with their putting in their tendrils ready to take over the seagrass cells. Then this one is really cool. This one, we added a nuclear stain. And what we can see in here is that there's a ton of LABI cells in the dead cells. So each one of those little tiny blue dots is a LABI cell that is inside and in there eating all the cell tissue of the seagrass. All right, talking about water residence time. We want to know more about LABI vectoring and transmission, so we want to find out how much LABI actually hangs out in the water as opposed to sticking on surfaces. We did this experiment here. It's an ongoing experiment. It ends tomorrow. Yeah, so we have 5 controls and 5 experimental tanks. We're taking daily water samples. We want to try and understand transmission a little bit better. We'll measure intensity and severity. And this is part of a much larger NSF project where we're making a model. The model will simulate the infection and progression of eelgrass wasting disease and focuses on disease control by environmental conditions. This is what our setup looks like right now. This is what one of the tanks looks like with some eelgrass in it. And this is my hypothesized graph. So basically, we inoculated it with we inoculated the tanks with the pathogen on day 0. And as soon as we turned on the water 24 hours later, the pathogen is going to start flowing out because it's a flow-through system. So it's going to start flowing out and flowing out. But at some point, the pathogen is going to take hold of the leaves and start to create lesions. So as they start to create lesions, they'll start shedding into the water. And hypothetically, the amount of LABI will go up in the water. Next steps on this one, we're going to extract DNA and RNA. We're going to run qPCR and reverse transcriptase qPCR. We'll do our analysis, and we'll get pathogen intensity. Another project that I'm working on right now is we have eelgrass samples from Alaska all the way down to Southern California. And we're doing a bioinformatics project where we're next-gen sequencing and running a bioinformatic pipeline to identify genetic variations in strains and in virulence. And we're doing So in conclusion, oyster sanitization treatments did work to reduce severity. So we highly encourage the seagrass industry to treat their oysters before they ship them off. The presence of live oysters didn't reduce LABI, but we had background infection, which was a confounding variable. We found new insights of LABI infection behavior, and we want to understand water residence time and how it affects disease transmission. And with that, I want to thank my PhD lab and my current lab for everything they've done. And I also want to thank all of our funding sources and stakeholders. That's it.
Do we have any questions? For those of you online, if you put them in the chat, I will read off questions.
Are there good surveys of the distribution of the wasting in eelgrass population on the coast? Variable in different? See any patterns? Tell you anything about trends?
I don't think there are quite that extensive of surveys. I know that surveys are being done right now in the state of Washington at very, very early stages. But I've come.
So all of the sites that showed showing the 18S sequencing data, they also conduct surveys as part of another NSF project. Jay or Drew, who are online, probably tell us more about this. I can tell you in Washington State, a lot of people across sites. More questions? I guess if no one is going, oh, someone.
Hi. Really interesting talk. Lots of different topics that I'm not super familiar with. So, how far offshore were the orca buoys that you did the
How far
climate?
off for what?
How far offshore were the buoys, like from
From each
shore?
other?
Or yeah, from each other, but also like so thinking about like the temperature, the difference you saw in temperature between 5 and 20, and where you might expect the sort of like thermal climb with the open ocean versus a little bit short. So that.
You're just wondering how far away they were? Or?
Yeah.
So I would say probably about 40-ish miles from each other, maybe more.
I'm coming on you. Sure you want that?
Nice.
Super cool. As you predicted when our talk earlier, the microscopy work. Oh, so cool to see those visualizations. How quickly were they moving? How quickly did you see some of the, both like when it's just in the water, but also when it was on the fieldgrass? You get an idea of how fast
Yeah.
Labby can move. And yeah.
Yeah. What you saw was probably, we did an overnight trial, but I just took like kind of a small snippet of it. So what you probably saw there was probably 2 hours worth. Yeah. They move pretty fast.
Yeah. It like, do you think it would be possible to extrapolate that grass bed or water? I'm
Like,
just in terms of like how quick that spread or how it's modeled, or do you think that's somewhat one?
yeah, we were thinking about that. We can, since we have a timestamp on them, we can actually see how fast they spread, and it should be some pretty easy calculations. Yeah.
So we have a question online, and I'm going to add it a little bit here because I think there's a little more detail. So in your laboratory trials, bring fieldgrass in from the field, are there incubation periods that ensure the eelgrass are under good environmental conditions? So how do we treat the eelgrass that comes in from the field?
So we put them in a low salinity bath, and that hypothetically kills all the labby, but the eelgrass are pretty unaffected by it, and we do that for 24 hours. It doesn't get all of it, as we learned, but it gets most of it.
I'm getting my workout.
Thanks. I actually just have this really basic question because I don't know anything about the lab. Does it, or I don't know, like, but does it feed on other things or get, nutrients or it's from other things other than eelgrass, or it's really just eelgrass?
Question. I know the answer to that.
And another question I have, which I from the East Coast, I can't remember, is it known whether it's native or its origin? I thought there were some questions about, whether it was non-native areas. I just can't remember the background.
Yeah. I think, was it introduced? Oh, no. It originally started in Europe, came to the East Coast, and is now on the West Coast. So it wasn't quite as big of a concern 20, 30 years ago.
So for the first part of your talk on shellfish, you looked at how OA was affecting the periostracum, and I was wondering if you saw any effects on the middle prismatic layer, the inorganic layers, because it seems like that might be more vulnerable than the outer organic periostracum.
Yeah, we measured total area or total shell density, and we didn't see any differences. Just the periostracum.
So I will ask you a question and then I'll read off the next one from online. So, as regulator for the state of California in charge of imports, would you, to assure that we're not transferring LABI, which method would prioritize? I know that the data was variable, or would you suggest using methods?
Yeah, the data was variable. It was really hard to tell because they all just kind of had kind of messy data. I would recommend one of the three because they all seem to be helpful.
So from online, the question is, how far out go when you did pH level experiments on shellfish that were under stress from severe temperatures?
Far out.
I'm guessing, how long was your experiment?
Oh, which is
The one with pH?
the first one?
Yeah.
That was only a 6-week experiment.
Now, to follow with that, those shellfish were not pre-stressed, correct? Those shellfish were normal,
Wild.
happy shellfish, and you didn't use any pre-stressed shellfish in your experiment. One more question? What?
Hi, awesome talk. I have a question about the sanitation method that came up right now. Has it been tried, or would you ever thought of like doing like two of the sanitation methods? Is that like too much for the oysters? Like maybe like bleach them first and then put in liquid right after, or vice versa?
That would be a really good idea. I think it's a matter of money for the shellfish growers. They only want to do one thing, but that would definitely be advisable.
I would also like to say that was 8 tanks the 3 of us were working with, so that would be that's undertaking. If there are any other questions? So please come to the South Lounge in about 20 minutes for refreshments, and join me in thanking Lindsay for a fantastic seminar.